Hello everyone, and welcome to the IBH webinar series. I am Yunqing at the James Hutton Institute, and today is my great pleasure to introduce Professor Klaus Pillen. Professor Pillen, he studied agriculture sciences at the University of Bonn. He received his PhD in plant biology at Ludwig Maximilian University. Then he became a postdoc fellow of the Human Frontier Science Organization, which supported his study in tomato genetics at Cornell University. And afterwards, he has worked as a researcher and group lead at University of Bonn and also Max Planck Institute Cologne. Since 2008, he has been a full professor of plant breeding at Martin Luther University Halle. Since last year, he's also the president of the German Plant Breeding Society. Pillen's major research interest involves molecular breeding, genomics, and quantitative genetics of barley and wheat. Additionally, he uses AI to combine genomic data, spectral and climate data to model and predict plant growth and plant performance utilized in plant breeding. Today, he will give us a talk about his work on wild barley alleles to improve stress tolerance in elect barley. With that, we want to welcome Professor Pillen.
Okay, thanks a lot for the introduction, and welcome to my presentation on exploring wild barley alleles to improve stress tolerance in barley. During my talk, I will first introduce our barley nested association mapping population HEP25 or Halle Exotic Barley 25 containing new alleles controlling plant development and yield performance under stress. We continue with a global study with a subset of the population called HEP yield to study grain yield under 4 abiotic stresses in a worldwide field trial. Then third, wild barley alleles interacting with the rhizosphere microbiota are shown, also related to the HEP25 population. Fourth, we show the studying of wild barley alleles, wild barley allele effects of one particular gene, early flowering 3, in growth chamber and field trial experiments. We continue with the wild barley alleles controlling culm and spike architecture. And finally, I will introduce you to the current status of studying the allelic diversity in the new wild barley pan-genomes. With this, I start with, let's say, first introducing the population we built during the last 10 years in order to get a hand on the exotic diversity present in Hordeum vulgare wild barley. And that's done with the HEP25 population by first crossing Barke, a German spring barley cultivar, with 25 wild barley Hordeum spontaneum accessions here indicated with colored bars indicating 1 chromosome pair. Barke is in black, and so if you cross them, you have heterozygote F1s. And then we did 1 round of backcrossing in order to reduce the average load of the Hordeum spontaneum wild barley in each of the lines we produce. And after that, we did 3 rounds of selfing in order to increase the amount of homozygote loci present in the population we built in this way. So the total population in BC1 S3 generation is made up of this 25 crosses. We call them then 25 families. In total, these are 1,420 num lines, and they are expected to segregate 72% for Hordeum vulgare homozygote, 6% for heterozygote in the BC1 S3 generation, and 22% of Hordeum spontaneum homozygote. The advantage of, let's say, this concept is that we have, let's say, a low amount of heterozygotes still present in the material, which can be used for validation experiments. I will explain to you later on. And what you see to the right is the origin of the 25 Hordeum spontaneums. They are mainly originating from the Fertile Crescent, with some extra accessions coming from Afghanistan and one from Tibet, which is actually not a Hordeum spontaneum, but a Hordeum agiochiton species. The population can be used either as a complete set of 1420 lines for genome-wide association studies, or in a subset which is easier to manage, made of 400 lines. We call it HEP 400. And today I will present one extra set of lines, HEP yield, which is made up of 48 lines, segregating more or less one-to-one for 4 particular genes of interest to us. The HEP 400 and the HEP yield are selected for non-brittleness of the rachis and for good thrashability, whereas the total population of 1400 lines also contain exotic alleles at the brittle locus and at the thrashability locus. The material, the complete population was genotyped with first with a 9K SNP chip, then with a 50K SNP chip, and in addition with exome capture data in collaboration with James Hutton, containing 400,000 SNPs in the exome capture data. Currently, or let's say in addition, we have full genome sequences, first of Barke, which is part of the Barley pan-genome publication from Yaya Cody et al. 2020. Then 5 HEP donors were already part of the wild barley pan-genome, Yaya Cody 2024. And the remaining 20 of the 25 donors, the remaining 20 are currently under development for a complete set of HEP 25 pan-genome. What we can do is indicated here. So let's say usually we do a complete genome-wide association study with a total of 1400 or 400 lines based on phenotyping new traits and using the genotype data indicated before. Or as an alternative, we first, in collaboration with partners all around the world, first do a screening of the 25 HEP donors to look for variation compared to the recurrent parent Barke. And then after the screen is done, we do a GWAS restricted to only those subfamilies where the donors have interesting phenotypes. So this is an alternative for the second one. And the third is validation experiment where, let's say, we use the concept indicated or published by Bergelson and Roux called heterogeneous inbred families, where, let's say, out of the HEP25 population for each SNP locus or QTL locus, we could identify the 6% lines which are heterozygote, and out of these heterozygote lines here indicated in this box, we can select, due to 1 or 2 rounds of selfing, select homozygote lines, segregating after 1 round. It would be 1/4 homozygote blue and the other homozygote red, and 2/4 half of the 2/4 are still heterozygote. And so these fixed lines with a fixed background already in the original HEP line, and then in addition segregating 1-to-1 for the locus of interest, the QTL, can be used for validation with, let's say, only 2 lines in a couple of replications. And this is called a HIF pair, and we will see that later on. So I mentioned that we did quite a number of studies so far and published those. These are 35 publications listed here, and they can be downloaded there. And out of those, I will now present examples of how we carried out in collaboration with collaborators from all over the world during the last 10 years. One study is on plant development, the second is on drought stress tolerance, the third is on heat stress and yield-related traits to early flowering 3. And then one is about phytomeric growth, and the final is an update on barley pan-genome sequencing results for HEP25. In order to start with the first example, I am showing this genetic model of flowering regulation. Here you see independent pathways indicated in red, so external cues which are used, or let's say which are setting the state for flowering. That could be a photoperiod, could be circadian clock, vernalization, as well as an autonomous and a gibberellic acid pathway, giving rise to, let's say, expression of the APETALA1 gene, which then in the shoot apical meristem changes or induces a transition from vegetative to generative growth. In cereals, here I indicated, let's say, easier, reduced schedule of genes involved. So the photoperiod is shown here with PPD H1 in case of barley, or PPD1 in case of wheat, in inducing constants, and constants is transferring the signal to VERN3 or the FT homolog in Arabidopsis, and the VERN3 protein then is transferred from the leaf to the shoot apical meristem, and there it's inducing APETALA1 as shown here. And in addition, in regard to the vernalization treatment here, it's shown that VERN1 is induced by cold and inducing VERN3, but at the same time repressing VERN2, which on itself was repressing VERN3 before. So if you want to see what's the situation in barley, here are indicated in green the some of the most important genes involved in flowering regulation, and in blue I indicate the counterparts from the Arabidopsis field. And those genes I highlighted because those are, let's say, genes we could find in our initial studies on flowering and plant development regulation in the HEP25. That is shown here. So that's the first experiment published by our group with Andreas Maurer as a PhD student at that time. And what you see in the circus plot is that, let's say, the highlighted yellow circle indicates the effects of particular regions of the chromosome. Here in the center, you see the 7 barley chromosomes. The gray lines indicate SNPs, which were studied in HEP25. And then for each of the circle, you see how the boxes indicating that there are significant effects on the trait, in this case heading, from a particular QTL region defined by an SNP next to it. So what we could see is that, let's say, we have 4 major wild barley alleles giving rise to early flowering or reduced time to heading. This is early maturity 8, which is the F3 or early flowering gene in Arabidopsis, the PPD H1 gene, which is a pseudo response regulator in Arabidopsis, central radialis, which is terminal flower locus in Arabidopsis, and semi-dwarf gene on chromosome 3, which is a gabberellic acid 20 oxidase. And at the same time, we could see that we have 4 further major genes controlling flowering time in the other direction. So the exotic allele in this case it gives rise to late flowering. This is one QTL still not identified yet. And then the 3 vernalization genes, VernH2 blocking flowering development during winter, VernH1 increasing after vernalization and giving rise to the signal, for FT1 VernH3, which then transmits the signal to the meristem, apical meristem. In this study, we extended from flowering time to the whole plant development. That's indicated here. So what you see is that for the other traits shown here, shoot elongation, heading, ripening phase, maturity, 1000 grain weight, and plant height, we could see that, for instance, for PPDH1, all traits except 1000 grain weight had effects, and in all cases except ripening, it was, let's say, reducing of the trait, so reducing plant height, for instance. The same is true for semi-dwarf gene 1, that we could see effects for all studied traits in this case, and again for VernH1, the petala gene, and VernH3, the flower locus T gene. So that was the first experiment. Then those 4 highlighted major genes controlling plant development we wanted to see not in small field trials with double rows as shown in this case, but in real yield plots to see how these plant development traits resulted in yield and in yield components under stress and control conditions. And for this, we had a worldwide field trial conducted in collaboration with partners at different stations and let's say carried out by Matthias Wickmann as a PhD student. The studies took place in Dundee, in Halle, in Al-Karak, this is Jordan, in Dubai, United Arab Republic, and in Adelaide in Australia. And the stresses applied are in Dundee and Halle nitrogen deficiency, so lack of nitrogen fertilization, in Al-Karak drought, in Dubai salt stress, and in Adelaide drought stress again. And to cut a long story short, I only show now the results for one of the genes under study with a HEP yield set of 48 lines. This is PPDH1. So we look for the difference between the Barke allele present in those 48 HEP lines compared to the presence of the exotic allele for PPDH1. And here I only show results under stress because the results under control conditions are very similar, but less expressed, less strong as under stress conditions. The 0 line indicates the performance of the Barke PPDH1 allele on heading, height, plant height, number of ears, number of grains per ear, 1000 grain weight, harvest index, and yield. And then the deviation of from the Barke allele is shown here as a box for the exotic allele. And what you see is that, let's say, we try to see first if heading has an effect on yield under the assumption that PPDH1 is causing this one part of the effect. And then we have these yield components to explain how PPDH1 is acting on yield. And under Dundee and Halle conditions, so long days during plant growth, we see early flowering, extreme early flowering, so 8 to 10 days earlier flowering. Here under nitrogen stress. But as said before, it's also visible under control conditions. With this, you see an increase in number of years here of the PPDH1 exotic allele, but a reduced number of grains, giving rise in combination most likely to part of the yield reduction, which is present if the exotic allele is present. So that means under Dundee and Halle conditions with long days, you should not select the exotic allele of PPDH1 because the final yield is reduced. If you now look to the same PPDH1 lines in Al-Karak, which is in Jordan, so much closer to the equator, then still you see the effect of early flowering of PPDH1, but now the effect on yield is the other way around. So what you see is that we have an increased 1,000 grain weight, significantly increased 1,000 grain weight, no effects on number of ears and number of grains per ear, and this 1,000 grain weight effect most likely give rise to higher grain yield under this condition. So it's like an escape mechanism that, let's say, with early flowering PPDH1 wild barley allele, you emit the strong heat and drought at the end of the growing season. Now, if you go further, I'm showing Adelaide, then there you don't see PPDH1 effects on heading, but what you see is that you have, let's say, an increased effect under stress, under drought stress in this case, on number of years, although this is not a significant effect. You see a significant effect of the PPDH1 exotic allele on harvest index in Adelaide, and this results, these two effects most likely results in the grain yield effect on from exotic PPDH1 alleles in that material. So indicating that under Australian conditions, even if PPDH1 has no direct effect on heading, it's still useful in order to increase yield under drought. This experiment was meant for, let's say, looking for applicability of exotic plant development genes present in HEP25 lines. And for this, the 48 lines we tested are shown here from the situation in Al-Karak in Jordan under well-watered conditions and under drought stress conditions. And what we could see that compared to the local Czech cultivar rum, which had under well-watered conditions something like 3.7 tons of grain yield and 2.6 tons of grain yield under stress conditions, or under drought stress, that, let's say, two lines, HEP lines, 0.5-43 and 10-173, they are similar under well-watered conditions here and there compared to rum, but under stress conditions, they are even better than rum, indicating that, let's say, HEP lines could be used in order to improve yield under stress conditions in particular regions, in this case, Al-Karak. I have to mention or confess that under high-yielding conditions in Scotland and in Germany, we did not find a HEP line which is, let's say, on the same yield level as Barke. The third example of using HEP25 is shown in a collaboration with David Bulgarelli from James Hutton. So this study was conducted by David and Carmen Escudero Martinez for looking to gene shaping the microbiota composition in the HEP25 rhizosphere. So in this case, so microbial effects can be expected above and below ground on plant growth promotion, improved stress tolerance, induced systemic resistance, and enhanced nutrient uptake. And in order to characterize the microbiota composition in the rhizosphere, David and his group used, let's say, the standard conditions of sequencing 16S RNA and internal transcribed spacers between the RNA genes in order to find amplicon sequencing variants. And this was applied. And what they could show is that, let's say, in the 25 wild donors of HEP25, they studied in a, let's say, selection experiment. They found, let's say, enrichment, a significant enrichment of a particular amplicon sequencing variants derived from proteobacteria and acidobacteria, shown here. For instance, one particular donor called Hordeum ID or HID 144. And on the other side, they found, let's say, an enrichment of significantly enriched bacteria species in the cultivar Barke on the other side. So they selected HID 144, which is present in this allele is present in the subfamily HEP15, made up of about 50 to 60 HEP lines. They conducted a QTL experiment, and what they could show in this circus plot is that on a family, genus, and amplicon sequencing variant level, they found an enrichment of protobacteria and acidobacteria at locus on chromosome 3H, which they termed QTL for rhizosphere microbiota composition. So that was studied in more detail than based on the HIF concept, so nearly isogenic lines or HIF pairs. This is a HIF pair where, let's say, you see that, let's say, in two areas, you have a variation. So blue indicates Barke alleles, SNPs are present. Yellow indicates the exotic barley alleles from HID 144 are present. And what they could show is that in this region defined here, in this region, that there are several, let's say, SNPs showing a significant higher gene expression of the genes next to the SNP in either the Barke alleles, for instance, the NLR-like gene, or in the wild barley allele, which is in this case is an undefined gene, so not defined gene yet. The idea was that we may be able to see that these QTL directly shows effects either on yield-related traits or on metabolites, but that was not the case. Here I'm only showing the results for the yield trials. In 4 replications, what you see is the Barke control, and then the 2 nearly isogenic lines. The blue one covers the Barke allele at this locus, and the yellow box plot indicates the line 124-52 covering the exotic allele. And what you see is that, let's say, you see a short increase in replicate 3 and 4, but this is not significant, indicating here by the same letters. So we could not see a direct effect of enrichment of particular microbes in the rhizosphere of the barley plants if the exotic allele is present. Then fourth experiment I'm showing is given here. So that's related to one of the major genes, F3, early flowering 3, or MUT-A. And that's a member of the circadian clock, and in particular of the evening complex, which, let's say, is expressed not during the daylight, but during the night. So when light is gone, here you see the peak of its expression, and below in this publication it was indicated how these gene works. So the ellipses are indicating proteins, and F3 is, let's say, a repressor of the PPDH1 gene. Remember, pseudo-response regulator 7, that's the PPDH1. And it's also, let's say, the compound F3, F4 and lux, they are, let's say, blocking the lux expression. So now we wanted to see how this is related to effects on yield components. For this, I recall the HIF concept, where, let's say, we have two final HIF homozygote lines, which only segregate, ideally only segregate at the locus of interest. So in that case, the F3 locus. So the one to the left might be the one having the Barke allele at the F3 locus, and the one to the right that has the cultivar, the exotic allele present at this locus. Now, what Zihao Zhu and Marcel Quint at the Halle University in this collaboration did is a growth chamber experiment. And I didn't mention that F3 also, let's say, is in use or is showing effects under elevated temperatures, rather 20 degrees in a growth chamber. They used 28 degrees. So the two alleles, or let's say the two HIF pairs are showing here. To the left, under 20 and under 28 degrees, they are showing the Hordeum vulgare, so the Barke allele, and to the right, the same temperature procedure for the HIF line, which has the exotic barley Hordeum spontaneum allele present in the HIF called 10-190. And what you see is that under in the HV allele, it's a slowing down of the plant development. For instance, here ZOX 1.5 after 19 days is reached at 20 degrees, and at 28 degrees it's reducing. The opposite is the case if you have the exotic allele. Then you see at the same time of day that you have, let's say, already first the exotic allele itself is increasing plant development, so you reach already ZOX 2.0, and if you increase the temperature, then it's even further increasing to 3.5. That is shown partly here. It's also compared to Bowman and F3 mutant in Bowman. That what you see is that blue and red are indicating the two temperature regimes, and that for the 10-190 line, the wild allele is reducing, as shown here, is reducing days to heading. Now, as I mentioned before, in this growth chamber experiment, we wanted to see a first indication of how that is related to yield-related traits. We did not see. Now we are focused on the effects of elevated temperatures in the exotic allele of the HIF. There we see that, let's say, it has no effect, elevated temperature has no effect on grains per plant, but it has an effect on grains per spike. It's reducing the number of grains per spike. The same is true for 1,000 grain weight and also for the total grain weight. It's a reduction, a significant reduction. The same is true here in the sibling line, the near-isogenic line, the HIF allele with the Barke allele, that you have a strong reduction in total grain weight, much stronger than in case of the exotic allele, but it's the same effect, reducing 28 degrees is a stress, and it's reducing the total grain weight. Now, from the growth chamber experiments, we wanted to extend this to field experiments, and these are conducted in Halle in our experimental field station. Here is an aerial view conducted by Tanya Zahn, my PhD student at that time. And here what you see is the result of, let's say, the 2 years of field experiments, 2019 and 20, with a number of different HIF introgression lines or HIF pairs. And as shown before in other cases, the 0 line indicates the level of the trait of the HIF line containing the Barke allele, and then the deviation is shown in the exotic allele. So for shooting, for instance, here are the abbreviations. Shooting as a first indication of plant development. You see in one line, or one HIF pair, I already showed before that you have, let's say, early flowering of the L3 allele, early flowering by 4 days earlier compared to the exotic allele, to the Barke allele at this locus, if the exotic allele is present, and that continues during heading and also maturity, that the exotic allele present in exotic allele of L3 present in this HIF is reducing plant development or speeding up plant development. We had no effects on plant height, but effects on number of ears per square meter, which is increased, but unfortunately grain number per year is reduced, no effect on 1,000 grain weight, and no effect on yield. So let's say due to the conditions in these 2 years, 2019 and 2020, the exotic allele of L3 induces early plant development, increasing the number of years, but reducing the grain number per years, and it's the similar grain yield present in both HIF lines shown here. What is going on with the expression of the genes under interest? What we could see is that F3 is increased during the night as expected. It has no difference between the two HIF lines. So the red is the Hordeum spontaneum and the black one is the Hordeum vulgare alternative in this HIF pair of the F3 locus. It has no effect on PPDH1 between the two lines, but in the Hordeum spontaneum line, you have, let's say, an increase in flowering time, most likely giving rise to the early flowering. And this is also true for VernH1, so Apetala1, which is the final signal for inducing of flowering buds. Okay, so then the question is, what is the difference between these between the allele present in this HIF line compared to the Barke line, and that's shown here for all of the tested HIF lines of ELF3 from different resources. And this is an amino acid position. And here you could see that this particular line, which is present in Hep family 10, where the donor of the exotic allele is HID-102, it has the same amino acid sequence as Barke, with one exception, and that's in the C terminal of the protein at position 669, where you have a transition from tryptophan to glycine, from Barke to the exotic allele, and in collaboration with Panagiotis Kastritis at the Protein Center at Halle University, we could show that this is related to, based on an AlphaFold testing, to a conformational change in between the Barke and the HID allele of ELF3. So that was a nice example that we can relate phenotypes to a single SNP or amino acid substitution. And the next example of using Hep25 is shown here, and that's a collaboration with Yongyu Wang and Thorsten Schnurbusch. And Yongyu did very precise measurements of spike architecture. And here are shown, for instance, first I should indicate the phytomere is visible in the vegetative growth phase, so the internode and the node, giving rise to new leaves appearing during plant growth. And the same concept the group also uses for the spike architecture, where, let's say, you have a node and an internode in the rachis. So in order to characterize now the performance of spike development, you have the potential rachis node numbers Yongyu was counting, and then a reduced number in the final rachis node number at, let's say, later steps of the plant development, for instance, during heading. And the same Yongyu applied to, let's say, the stem growth, where he's calculating the Kulm node number. And what he could show is that, let's say, in the Hep25 population, he used about 250 selected lines out of the total number of Hep lines. He could show that he had in the exotic alleles proximal rachis internodes increase, and the distal Kulm internodes, which are elongated in wild barley phytomeres. That's shown here, and there it's easier to see because you have the introgression lines compared to the second population made of domestic 6-row spring barleys. He uses in the two populations, so the DS6 population on one side, and in the introgression lines, which is a subset of the Hep25, made of 246 lines. He did GWAS for 14 traits shown here to the left, and he could show that in the domestic population, he found 8 QTLs in the introgression lines, or Hep25. He found 12 QTLs shown here and indicated by names, and these are most of them are the same names as we found before for plant development. And here it's for particular, let's say, traits related to the phytomere extension. And Then in the same publication, Yongyi indicated one example how this action is going on, and that's he selected two of the major genes controlling the spike and stem morphology. And these are PPDH1 and the semi-dwarf gene, so gibberellic acid oxidase gene on 3H on 2H, sorry, and he did not see. I mean, he saw individual effects of the two genes for the distal, central, and proximal culm internode length, and also for raches, so spike internode length, and for days to heading. But he was in particular interested if he could also see interaction or epistatic interaction effects. For the distal culm internode length, he could not see in interaction effects. So those lines which had, let's say, exotic alleles present at semi-dwarf 1 and PPDH1 at the same time, they are shown here in the black box. Those which have only one exotic allele, either present at PPDH1 or present at semi-dwarf 1, are shown in the gray and in the dark gray boxes, and they are normalized. So the 0 level is the control of two alleles at PPDH1 and semi-dwarf derived from the Barke in the same subset of the population of 247 lines tested in total. And so in regard to an additive effect, interaction effect, he could not see this on the distal culm internode, but he could nicely show this in the central internodes. So central internodes are, let's say, in this part, and the proximal are at the base. There again, you see a strong interacting effect, giving rise to higher levels if both exotic alleles are present. It's also the case in the raches internode length, and it's also visible in the days to heading trait. So that was a nice example that, let's say, based on the study, you could show that exotic alleles have, let's say, direct effects on shaping the architecture of a spike. And now the last example I'm going to show is given here. This is the barley pan-genome, the current version published by Yaya Cody in 2024. This is made up of 76 accessions containing 17 elite cultivars, 36 land races, and 23 wild barleys. And the wild barleys include already 5 Rhodium spontaneum donors of the HEP25. Here you see a diversity of the material. Here you see one example shown in this publication of a translocation event in one of the donors, HID 055, compared to Barke and compared to the other 4 donors. And here you see high C map where, again, based on LD decay, you see a strong LD indicating the translocation between 2H and 4H of the wild barley 055 compared to Barke. The question in this publication was if we have enough domestic accessions for the pan-genome to cover most of the genome space or gene space. And you see here already that with increasing the number of accessions further, you reach already a plateau coming close to 600, 650 megabase of the size of a single copy pan-genome. Whereas in the case of wild barleys, there is more diversity present that, let's say, based on the 20 3 wild barley accessions, we are still in exponential growth curves. You didn't reach the plateau yet. That is for us an incentive to continue with sequencing the remaining 20, the missing 20 donors, exotic donors of the HEP25 set, and that is conducted by Thomas Schmutzer in my group, in the subgroup of bioinformatics. Here you see first, let's say, pre-trial where Thomas compared Barke and one wild barley FT11, and what he could see is that, let's say, most of the structural variants are based in the internal part of the genes, and these are either insertions, duplications, or deletions, and that he conducted already for the 25 populations 25 accessions of HEP25 in total, so the HIDs. And here you could see that, let's say, the concept of differentiating between genes which are in the core, so they are shared by all, compared to they are in the shell, they are shared by some accessions, but not by all. And finally, the cloud, which are, let's say, private, which are only present in one of the genomes, can be, let's say, visualized now compared to in the spaces of the genes that, let's say, you have a high number of cloud or private structural variants present in the break ends, indicated here in purple, and you have a high number of core, or we call it soft core, structural variants which are present in insertions of genes. In total, structural variants are present in 37% of the annotated genes, and 340,000 accession specific structural variants are private, so they are only present in one of the HID accessions. This is almost the last slide that's showing, based on the sequencing of the 25 lines, some were already present in the barley pan-genome. They are indicated in brown. The cultivar is indicated in blue, and the green ones are the 20 additional wild barleys present in HEP25. And based on the phylogenetic tree, I, let's say, split it in 5 groups, color-coded groups, indicating that closest to Barke are HID3 and HID 380. This one, 380, that's the agriocriton. It's unclear to us why HID 003 is even closer to Barke compared to agriocriton, but this is Podium spontaneum line. In total, Thomas Schmutzer could find approximately 50,000 annotated protein coding genes in each of the HID accessions, and 16% are core genes in the core, so they are only present, no, sorry, they are present in all accessions at the same time. 53% are in the shell, and 29% are in the cloud or are private genes. They are shown here. Here, sorry, and they indicate that on average, 1,000 novel genes are present in each of the wild barley accessions. Now, here you see a map of the 25 sequences, chromosome level sequences of the AB25 donor in addition with Barke. And this is the Barley 1K accession, Hordeum spontaneum accessions with the name FT11, which I showed here already. And what you could see is that, let's say, you have, let's say, not only the in the publication already shown translocation event in HID 055 on chromosome 2H and 4H, but you have, let's say, in the total set 5 major chromosomal translocation events present in the AB25 population. And in addition, we also identified 5 major chromosomal insertion present. So they are indicated here that in this rectangular white triangle, that there is a piece of chromosome missing in one line, which is present in the other line or other accessions. And they are involved either in chromosome 2, insertions, deletions, or in chromosome 5, insertions, deletions. Yeah, with this, I'm coming to the end, to the conclusion. The barley, the wild barley nested association mapping population is a rich reservoir for exotic alleles controlling plant development and yield formation. We also could show that global field trials of Hab yield, the subpopulation, revealed striking variation of flowering time genes on or effects of flowering time genes on plant development, yield, and yield components under stress. The wild barley QDL alleles control rhizosphere microbiota composition, and wild barley allele of L3 promotes early flowering at elevated temperatures. The additive PPDH1 SD semi-dwarf 1 interaction coordinates phytomere node initiation and internode elongation. And last but not least, the available wild barley pan-genome promotes allele mining, supporting barley breeding and barley research. Yeah, with this, I'm at the end. I presented data from our group in collaboration, let's say first from our group, from Andreas Maurer, Thomas Schmutzer, Yvonne Pöschel, Matthias Wickmann, Tanja Zahn, Zihao Zhu from the Quint Lab, and collaborations with Niels Stein, Martin Marscher, Thomas Thorsten Schnurbusch, Yongyu Wang, Michael Baum, Robbie Warr, and David Bulgarelli, and others which I could not name on this list. Yeah, with this I finish. Thanks a lot for listening, and I'm ready to take your questions.
Thanks, Claude, for the wonderful talk. It's a really great and valuable resource that supported a lot of different projects. If any of you have questions, and please put it in the chat box and I can read it for you. Or if you raise your hand, we can unmute you so you can ask your own question. So far, I don't see any questions in the chat box. Maybe I can ask the first question. So these wild barley lines, they're selected to study stress tolerance. I wonder if there are any prior knowledge of these lines. Are they really resistant to biotic or abiotic stresses, or this is like an assumption that the wild barley are generally more tolerant?
No, I mean, my feeling is that so either it's an escape mechanism, so related to those genes which are controlling plant development. So early flowering might give rise to reduced stress. That's the case for drought, perhaps also for heat, but definitely it's not the case for nitrogen stress, which is very important for us in Europe, I would say, because at least in Germany, we have, let's say, legal regulations forcing farmers to reduce their nitrogen fertilization. So we are looking for genes which are, let's say, better using the reduced level of nitrogen in the soil. And that is nothing related to plant development, that's independent. So these are genes, for instance, related to root growth or uptake of ammonium or nitrate, and then, let's say, reducing, if it's nitrate, then it's genes involved in nitrate reductase, or then finally related to the uptake of ammonium into organic compounds, so GOGAT and other genes involved in taking up ammonium into nitrogen into amino acids. Yeah. So from this point of view, I would say it depends on the trait. If it's if you can talk about escape mechanisms or on genes involved in the metabolism.
I guess my question is, do we know that, for example, with these wild barleys, this parent that you used require less nitrogen? Is it something being studied, or is it assumption that they may need?
No, I mean, that's let's say the problem of the material is that, let's say, studying yield-related traits in the wild barley is not easy. So, I mean, you can do that with single plants in the greenhouse, but then, let's say, translation to performance in the field is not, let's say, 100%, because it's a different situation if you have single plants or if you have a stand of several hundred plants per square meter. So there you have competition between the plants. So and that, let's say, to do in studying the donors, wild barleys in plots in the field is not easy.
Okay.
Yeah. So that's why we that's why we preferred to first do crosses and then back crosses, so that only a low amount of genes are present in each of the resulting lines.
Yeah, a good point. So there is a question from Gang Guo. Do you want to ask your question by yourself, or you want me to read it? I think I will read the question. Thanks for Professor Klaus' nice talk. My question is, when we utilizing the favorable allelic variations of wild barley during breeding, how can we minimize the introduction of unfavorable genetic baggage that affects yield and quality?
I mean, that's not easy. I mean, our concept was that we did the back crossing. Actually, it would have been better to do 2 rounds of back crossing in order to further reduce the effects of deleterious alleles present in the exotic material. But let's say 2 rounds of back crossing would mean that you need to increase to double the population size to have the same number of haplines showing the exotic alternative allele to the Barke allele. And so that would be the problem, that let's say 1400 lines in our case, giving rise to 22% of lines with the exotic allele, that this is already, the 1400 is already big. So if we want to have the same power of detecting effects from a particular line, then we would need 2,800, and even the 1,400 was very difficult to conduct. So we had experiments in Halle, and then the second generation, we had experiments in Dundee and in other places. I indicated the Hep yield, but at the same time, those lines were tested in the whole population with 1,400 lines. And that was a lot of work. And it's much easier to reduce it to, let's say, the set of 400 lines, but it's always the same situation. It's only 22% which have homozygote fixed genotype present in the material. And so that's hard to be used. So if we would have bigger capacities, field capacities, and man power, then it would be much better to increase it to 5,000. The original NUM concept carried out by at Buckler's lab in case of maize, that was 5,000 lines. But I think they also stopped using it because it was not easy to handle 5,000 lines, usually also in 2 treatments, so it was too big.
There's a question from Ruth. Did you have any issues with outcrossing? If you did observe outcrossing, was it coming from some of the Hep groups or wild parents more than others?
Yeah, we didn't study it in detail, but we have one publication where Andreas Maurer compared, let's say, the performance, let's say the genotyping results in the original BC1S3 generation, and then I think it was 5 generations, selfing generations later. So what he could show in this publication that, let's say, usually either the Barke allele or the exotic allele was, let's say, more or less fixed in some cases. And that is only visible if you have, let's say, heterozygotes. I mentioned we have 6% heterozygotes. So those, they were 6% of the 1,400 lines were heterozygote in the BC1S3 generation. So if you look 5 generations of selfing later, the original homozygotes, they segregated more or less 1-to-1 if there is no selection pressure. But what Andreas could show is that, for instance, brittleness disappeared because we selected against brittleness just by selecting the plants from the field which are still on the stem, and the others were already on the ground. And in other cases, it was a different story, that the exotic allele was favored compared to the wild, the exotic allele was favored compared to the elite allele. So this material could also have been used to look for, let's say, if there is a higher outcrossing rate. But with SNPs, it's not so easy to indicate if this is, let's say, a mixture or if it's an outcrossing. But it should be possible. Yeah. But we didn't check this in detail. We were more interested in selection pressure due to propagation of the material.
Okay, next question from Davide. My question is on part 5 of the talk. Have you looked into what differentiates between the clades of the heath? For example, particularly particular GO categories emerging when looking at polymorphic genes among accessions?
No, I mean, David, no, we didn't look. I mean, that was, we left it to you, David, to have a closer look. No, yeah.
Okay, there is one, I think it's a comment. Add this. I don't know if you want to make the comments by yourself.
Yeah. Okay, so I mean, we are open for collaborations. That's our concept, that we are not, let's say, doing everything ourselves, but that we like to collaborate. So if you want to use the material, that's no problem. We have a number of pathogen resistance studies conducted in particular in Australia, where they found nice resistance alleles from wild barleys, which were already used. So, I mean, I would recommend to look to this PDF page I indicated in the introduction, where all the 35 publications are listed. You just have to click on each of the publications to directly go to the publication or the journal and see the results or the study conducted. And let's say if you have a new trait which is not already studied or which will be studied under a different, for instance, environmental condition or treatment, then it would be nice to use it. And let's say we are fond of the HIF concept. We didn't introduce the HIF concept, but it was for us a nice situation for validation experiments. So that, let's say, based on the 6% heterozygotes in the original material, which are now, after almost 10 generations of selfing, fixed, it's easy to pick two lines which are nearly isogenic lines, and they can be used as a case control experiment or mutant compared to wild type experiment, where you do not need to have, let's say, a big population. Our problem with the QTL is that we need more than 100 plants to have a trustworthy QTL study conducted. And then, let's say, the next step is validation. And for this, we reduce it to 2 lines in a high number of replications or different treatments then.
Okay, so I think others just said that it will be great to collaborate together. And I think that's the end of the webinar today. We want to thank Crosstalk again. And I hope to see you at the next IBH webinar. Thank you, everyone.
Thanks a lot. Goodbye.
Bye-bye.